This is the Help Me video, question 3 of the Core Concepts Key Assessment 3, paper 1. A bit of a mouthful. We are given a bit of blurb, 5 lines of blurb, 4 diagrams, and a describe and explain. Right, we have got an enzyme. So an enzyme found, so that's our A's, that's our enzyme. Okay, and it's involved in DNA replication. Okay, so the enzyme catalyzes the conversion of a molecule. So that's our enzyme of pyrophosphate to 2 phosphates. Okay, so this pyrophosphate is our substrate. So that's our substrate. And our product is phosphate. Okay, that makes sort of sense. We need another oxygen from somewhere, but I'm not gonna worry about that. So enzyme is substrate, and it also tells us molecules of phenylalanine, that one, and phosphate are also shown. Yep. And those are both inhibitors, right? So this is a inhibitor, and this is also, I'll do it a different colour because it's going to be different, and this is also an inhibitor. So confusing diagrams, I know. We've got an enzyme, substrate, and 2 inhibitors of this process. The enzyme, look at the shape. Now what we are expecting to find, we are expecting to find an active site. So active site on here. This is a tertiary protein, 3D globular protein. Possibly one of these 2 things here. Okay. It's given us 2 for some reason. And what do we need to do? Describe and explain why pyrophosphatase can only hydrolyze pyrophosphate, and the mechanisms for the 2 inhibitions. So our first area is why this enzyme can only catalyze this substrate, converting into a product. That's our first area. Then our second and third areas that we're going to talk about are how does this inhibitor work and how does this inhibitor work. The clue is as to the type of inhibitor, the shape of phosphate is very much likely to be the similar shape, not the same, the similar shape to pyrophosphate. You can see it's a phosphorus with oxygens around it, a double bond and so on. So that's a phosphorus with oxygens around it, double bond. So it's very similar shape. So one of the methods of inhibition relies on similar shapes also fitting into active sites. And there's another type of inhibitor, and if you look at this, then in no way could this be a similar shape to the substrate. It's a different shape, and they've almost given you a clue as to what's going on, the shape of this and the shape of that there. So this is the other type of inhibitor that doesn't fit into the active site. So 3 areas, and now you know what you're talking about. You're talking about how do enzymes and substrates, work together, shapes, and how is it catalyzed. How do competitive inhibitors work, and how do non-competitive inhibitors work? And you just need to put that together in a sensible way for 9 marks.